Welcome everyone to another ALK Talk. And I said this the last time we had a meeting, and that is go to your YouTube channel, search ALK Positive, and Summer will put it in this recording so you can see it. And sometimes the ALK Talks might not, give you much information that pertains to you today, but you want one day or another, sometimes you will get an ALK Talk that applies to you. So please go to our YouTube channel and check it out. And we're going to skip poll questions, and we're coming live from Chicago, where Summer Farman and Von Diaz are there, and Kirk Smith was there, and some other people from our board. And I want to go ahead and turn it over to Summer.
Hello, everyone. Thank you for joining us this evening. I am Summer Farman from Hershey, Pennsylvania, coming to you from Chicago tonight, live from ASCO. I was diagnosed in 2020, and I'm here with my dear friend from London and ALK Positive partner in crime, Von Diaz.
Hi, everyone. It's such a pleasure to be with you tonight. As Summer says, I'm over from London here to attend ASCO with an amazing group tonight. ASCO is the American Society for Clinical Oncology. It's the largest cancer conference that takes place here in Chicago, and we're pleased to be with you. We've had an amazing group with us, including Kirk Smith, our president, Nancy Prezati, our vice president, Ken Culver, our director of research and clinical affairs, Mark Muskovich, our medical committee member, and also Sadia Yakov, our project manager. So we've been in full force, and it's exciting to be here with you on what is the first day of ALK Positive Awareness Month.
And we have been celebrating ALK Positive Month, Awareness Month hard for the past 2 days, nonstop. It has been 48 hours, but feels like 2 weeks in all the best ways. Our feet looked like little sausages yesterday as we raced about the 44,000 people at ASCO. We had tons of groundbreaking meetings, kicking it off with our second annual Scientific Advisory Board dinner Friday evening, and then just 2 days of meetings. So we're so excited. We, in fact, just got off the shuttle bus to join you all here tonight. So last year was our first ALK Talk live from ASCO, and it again kicked off ALK Positive Awareness Month by highlighting and announcing our collaboration with Breakthrough Cancer. We had Jesse Boehm on then, and he dared to return again to update you all on the exciting progress they've made in the past year at Breakthrough Cancer. But first, before we hand it over to him, we have been working Ken Culver to the bone.
True. Exactly true.
It is true. He's been racing around ASCO on an electric bike, fun fact. So he has a message for all of you. Katie, roll that clip, please.
Good evening, ALK community. Ken Culver here. I have the privilege of being at ASCO in Chicago, where we're hearing all the greatest and most important new developments in cancer research. And what I'd like you to know is that we are here advocating for you for new treatments, for new diagnostics, for new opportunities for our patient community to take advantage of these great new things developing. This is the beginning of the ALK Awareness Month, and tonight we're privileged to have Jesse Boehm from the Breakthrough Cancer Group with us. As you may know, they're one of our most important partners that we've been funding because they're working on that all-important minimal residual disease. In fact, we know that most of you out there have minimal residual disease. And through the raising of funds this month during our Awareness Month, we're going to be making sure that new discoveries and new advances can be made for MRD specific for patients with ALK-positive lung cancer. So we hope that you'll stay on and listen to this great talk by Jesse and learn the advances that are happening and our goals and our determination to cure this disease. And thank you for the privilege of being here at ASCO to represent you and moving cancer research forward for you.
Thank you so much. And yes, it is proof, it is true that Ken has been pushing the needle and advocating for us. And it's amazing to witness, and I'm so proud to be a part of it. for our entire community. So let's get right to it. Jesse, thank you so much for being here this evening with us, and please take it away.
Happy to be here. Thank you. Can you see my slides? Fantastic. So it's wonderful to be here. I'm not in Chicago myself. I'm stuck back in Boston, but we are working very closely in hand with ALK Positive on a really exciting initiative that I first mentioned last year that tries to take a new approach to target ALK positive lung cancer through the power of what we call radical collaboration. This is bringing together researchers, institutions, technologies, computational biology, and new treatments together as part of one national team to tackle ALK-positive lung cancer. And this is a model that we at Breakthrough Cancer follow for a number of different cancer types. And we really believe at this moment in time, the power of radical collaboration is really going to transform multiple cancer fields. So let me give you just a brief reminder of what Breakthrough Cancer is all about. We're a foundation. We're not a patient-focused foundation as you are. We're not a lung cancer foundation as you are, but we're a foundation that is trying to build a new recipe for how to tackle cancer in the United States. And then I'll open up at the end with a few updates on what we've been working on in the last 9 months with this new ALK Positive team lab in partnership with ALK Positive. So this is the time to tackle cancer, of course, the same level of urgency in the United States and beyond as we felt for COVID during the head of the pandemic, the same level of radical interaction between academics and industry researchers and the federal government. The same number of patients are being lost every day from all the horrible types of cancer as were lost at the peak of the pandemic. And we need to continue to bring this level of urgency to each of our careers in our laboratories. At Breakthrough Cancer, we challenge researchers and our biopharma partners to think long into the future about what it's really going to take to transform the status quo. Not what's the next grant, what's the next activity, what's the next trial, but what's it going to really take to transform the field as we know it. And then what are the barriers preventing us from flipping a switch and achieving that ultimate ambition today? And then we work with partners to provide initial catalytic funding for, let's say, an initial 4-year period to prove that the new concept that a team has developed that will make it possible to overcome these barriers actually holds water. And if we can prove that the barriers can be overcome, we then have an entity, a team, infrastructure, technology to then begin to scale and begin to reach the ultimate ambition working together. And it's this same recipe that we've taken with our ALK positive lung cancer researchers that we've taken for multiple other cancer types. At Breakthrough Cancer, we also work on ovarian cancer, glioblastoma, a deadly brain cancer, acute myelogenous leukemia, a deadly type of leukemia, as well as pancreatic cancer, and now a new initiative quite soon in a new rare pediatric cancer. And while each of these cancers are really different, the types of goals that we uncover when we challenge researchers to think outside the box end up being fairly similar. How do we find cancer at its earliest stages, before diagnosis or in the minimal residual disease state, as we'll talk about more, and intercept at that most vulnerable, minuscule stage, and do this in a systematic way? How do we accelerate drug approval? By working collaboratively with our biopharma partners and learning from every patient's experience. Of course, we want to do experiments in mice and in cells, in dishes, but we really know that we need to understand why a drug is working in a patient and why it is not, and get all of that information, all of those samples, all of that detail back into the laboratory and then iterate quickly towards the next trial. So these are principles that transcend each of our team lab projects. We also bring this new social structure to bear on cancer research, which we've kind of playfully called radical collaboration. We're really thinking about how humans, researchers, work together in a new way. It's not my lab and my institution and my name in lights. It's our lab, our team lab, working together across the country. We work with investigators to identify a common vision of what hill we are trying to climb together. We think about leadership not through the lens of seniority or hierarchy, but as catalysts to uplift young researchers and move them forward. We think about how to align the incentives of many different types of researchers and build a shared culture where everyone really feels good about working together. And of course, we work very hard with the institutions. Of course, sharing samples and data across the country is really hard, but we work closely with the institutions to ensure that there's as frictionless an exchange of resources and information as possible. And this requires real investment in the operational groundwork underpinning science. And we found that this is very, very important in enabling these team labs to succeed. And so this is the concept. Breakthrough Cancer was founded with 5 cancer centers: MD Anderson, Memorial Sloan Kettering, MIT, Johns Hopkins, and Dana-Farber. But as you'll hear with this lung project, we've expanded to 9 cancer centers where the work stays at the center, the dollars stay at the center, and people work together, eschewing their institutional identities as much as possible. And data science and new technology is really at the center. 2 special types of technology, well, actually 3 special types of technology are being used, not only in this lung project, but in each of our cancer projects. The first type of technology is called blood biopsy, enabling a sample to be taken in a tube of blood and monitor the trajectory of cancer all along the way across treatment, allowing clinicians to pivot very quickly in real time. A second really exciting new type of technology is called single cell spatial technology. And what this means is looking at a piece of tumor under the microscope and every single cell, how it interacts with every other cell. This type of technology has only just become available to researchers in the last 4 or 5 years, and we're now trying to move this technology into clinical samples as quickly as possible so we can see at the cellular level if the drug's working, why, and if it's not working, why not. And across our whole portfolio, there are 9 clinical trials with almost 150 patients enrolled thus far. And the final technology we lean on quite a bit is machine learning and artificial intelligence. And that's very, very important for this ALK positive lung cancer project that we've taken on hand in hand with ALK positive. So let me turn to that. Patients, of course, are treated. These are MRI scans from an example patient with metastatic ALK positive lung cancer. This is kind of a conventional way of thinking about the clinical time course. You can see in the scan, the black is no tumor, and this little red arrow is showcasing a bit of tumor in the lungs where it shouldn't be. We identify tumors, we treat patients, and even though we know that in many cases there's residual disease shown here in the center remaining after the initial treatment, we cross our fingers and hope for the best, and we don't continue to intervene in a different way until the disease has progressed. And unfortunately, at that point, there are so many cells and often so much genetic complexity that it's very challenging to achieve the type of cures and responses that we're really looking for. So what we decided to do in ALK positive lung cancer is similar to what we uncovered in many types of cancer, is to challenge researchers to think about a laser focus on this minimal residual disease state. Don't just cross your fingers and ignore it and hope that it disappears or stays there forever, but let's understand it. Let's sample it. Let's analyze it. And let's build clinical trials that enable this disease to be treated when it's at its smallest and most vulnerable. And let's build new machine learning and AI technologies to predict which patients will progress and which will not. So that's the essential vision. And I mentioned this last year to this group that this team is tackling. So last September, with the partnership of ALK Positive, we launched this team lab. That's our kind of virtual name for this shared laboratory. It's got a catchy name that we co-developed with ALK Positive called the Powered to Cure ALK Positive Lung Cancer Team Lab. The RD is capitalized because of residual disease. And this is a new identity that this team of researchers strung across 9 institutions all now has together. You know many of these researchers. I'll just mention a few: Christine Lovely and Vincent Lam and Justin Gaynor, Jess Lynn, Alex Drillin, Mark Awad, Simon Heek and John Haymock, Alice Shaw, Roberto Chiarli, Valsamo Agnosto, Chris Love, and Aaron Schenk. You probably heard some of those names, and there are others involved as well, but these are some of the best researchers in the country now all working together collaboratively as part of one new entity. This team has tackled 3 goals in their first 9 months, and they're continuing to work on these goals. These, of course, are not 9-month goals, but these are goals that will take many years to fully decipher. The first goal has to do with using this new technology that I mentioned called spatial single-cell profiling to analyze these minuscule residual disease samples, go back and sample them, and look to see all of these cells and how they're interacting with each other. These samples aren't frequently sampled. These samples aren't frequently taken from patients, but the team, through a number of clinical approaches and other approaches, is going back and trying to systematically collect these tissues so that they can develop a comprehensive understanding of the biology and the immunology of this minimal residual disease state. And that'll tell us, we think, some important drug targets, some important reasons the immune system is allowing this tumor tissue to stick around. And while we won't have all the drugs right at the beginning that will go after what's discovered, we think this information will provide a powerful roadmap for drug discovery. And that's why inclusion of biopharma partners is very important at the beginning of this project. The second main goal is to enable patients anywhere to donate tissue and samples. And so this is the extension of a national direct-to-patient tissue donation program that Jess Lynn, especially at MGH, has established. We want to break down the silos of every cancer center collecting and storing its own tumor tissues and kind of competing with each other. We want all patients to be able to donate tissue so that they can be shared with as many researchers as possible. And this is just getting going. This will involve a direct-to-patient remote consent, the ability for mobile phlebotomy to go and collect blood tubes, and then systematic data sharing. A really important part of this is to figure out whether remember I mentioned earlier, these blood samples, blood biopsy could be used to analyze tumors as they grow. One question is, can we do that in the same way at a local cancer center if you collect the tube and go downstairs and grab it, versus a tube that was donated from a patient all the way across the country? Do we have the same level of precision to track and see this disease? And we think the answer is yes, but that'll be very important for the team to evaluate in the next couple of years. And the final, perhaps the most important thing that the team is trying to launch right now is to develop a series of clinical trial protocols so that multiple drug interventions can be used in this MRD state, so that patients, they've been treated initially, they've gone into residual disease. Can we use new types of treatments like a cancer vaccine or other new drugs targeting these proteins called GAP-TEAD that are really important to keep the tumor cells alive? So these trials are just getting planned and launched now. We've had a very collaborative and really exciting interaction with our partners at Moderna just yesterday at ASCO, and I'll just give a brief update on that in my final slide. So here's where we are. I mentioned some of this last year, but last year we were -3 months, I think, from launching the actual work. We're now positive 9 months. So 9 months is not an enormous amount of time to try to get 20 researchers across 9 cancer centers to share thousands of things and work seamlessly as one entity. But I do have to say it's going really well so far. This group is meeting bi-weekly. They're really working like one lab. I know that doesn't sound like scientific progress, but that's really important culturally to see the coalescence of these folks. They've already shared hundreds of thousands of medical images across institutions. You might have heard a lot about artificial intelligence and making predictions and why it's really important to have a lot of data. It's hard to do this one institution at a time, but because of this master collaboration framework that we helped to set up across 9 cancer centers, they can all share these images and this data very, very quickly. And that's really speeding up the development of new machine learning models to predict which patients might progress fastest. Just recently, The teams have shared this new residual disease tissue that they've sampled across multiple cancer centers, especially MGH and MD Anderson. And I was just told, I don't have the data in front of me, but I was just told last week they developed the first spatial transcriptomics data. Remember, that's this single cell, how every cell is talking to each other. And they didn't just use yesterday's technology, they actually used one of the most powerful technologies that's ever been developed that very few tumor types are being processed with. So I'm really excited myself to see what this data looks like. I really think it's going to point to some new drug discovery approaches. They're working very hard to evaluate which technology is the best for evaluating ALK positive patients in this residual disease state. You might think that this is a small amount of tissue. The DNA has to be released into the blood. Then the blood sample has to have that DNA in it. So you need a super, super sensitive technology to find that needle in the haystack. The team is benchmarking two approaches, one from a company that is called Guardant Infinity technology. Another is called Haystack, but they're trying to do a very systematic job of comparing and contrasting different technologies. And we're really looking forward to seeing that data. The final thing that I'll mention, maybe the most exciting, is around the vaccine trial. There's been really intensive planning, and there's been two very important steps here. One is solidifying a relationship with a manufacturing partner that is committed to making this ALK vaccine. For the science aficionados in the group, this will be a vaccine with the cytoplasmic domain of ALK, like the whole part of the protein that's looking inside the cell. And so it can be processed. And then the question will be, does that really stimulate an immune response in patients? And this manufacturing approach requires a delivery vehicle. Remember, when you got your COVID vaccine, it wasn't just, it was RNA in this lipid particle so that it could get delivered. And this, the best of these delivery technologies comes from our colleagues at Moderna. And so just yesterday, we met with our colleagues at Moderna, and they heard about this project, very excited about it, and they gave us kind of an official green light to move forward with this proprietary formulation. So it hasn't fully been announced yet, but we're really excited about this. This clears the remaining hurdle to finish the planning of this vaccine trial. And the timelines look quite good. And if all goes well, the first patients will be enrolled in this vaccine residual disease trial next year. This will be a trial involving the vaccine plus one of these third generation ALK inhibitors called lorlatinib. And it'll focus probably on about 60 to 70 patients that have received another ALK therapy, such as alectinib or brigatinib, and have progressed and are now eligible for this new trial. The details are still being developed, so I don't have all of them in front of me today, but we're really looking forward to keeping this community in the loop as this new vaccine trial launches. And the final thing I'll mention is we weren't sure when we launched this project whether we really could get 9 institutions, 20 researchers, all these technologies, all this data science firepower together. But I'm really pleased to see what's happened just in 9 months, and really excited to think about updating you next year because I think we're going to see some really, really amazing science coming from the power of radical collaboration. And the final thing I'll mention is just a big thank you to our partners at ALK Positive. As I said before, we're not lung cancer experts. We're trying to build a new collaborative structure, and it's really important for us to have the partnership of ALK Positive and of all of the ALK Positive patients who we know are really excited about this and want to participate in many different ways. So let me stop there, and maybe Summer and others be happy to answer any questions that the community has.
Jesse, thank you so much. There are so many exciting updates. Thank you for first giving us the background information again. It's been a year. So this was a great recap for us, and also an introduction to breakthrough cancer for those that have been diagnosed in the meantime, because we know that that is happening all the time, every day. There are new members that join this family that you never wanted to be a part of, but here we are. And it's so exciting to hear that you're looking to take this from the next step from TKIs, because TKIs are a Band-Aid for us, right? It's not if we progress, it's when we progress. So we so appreciate this radical collaboration approach and all that you are doing to take us to the next step, to keep us in the game and get to the cure. So thank you. Okay, one thing that was particularly exciting for me, as people maybe want to ask Jesse some questions in the chat, and then Yvonne, you can do a takeaway too. But I love to hear about the benchmarking that you're doing, the sensitive benchmarking. Does that mean, for those of you wondering, and myself, could that possibly mean less scans for us in the future? Right now, we're scanned every 3 months. Are you thinking that that's what that could potentially replace?
Certainly in the fullness of time, Summer, that would be something quite exciting. You could imagine getting a blood sample maybe once per month or even more frequent and being able to see the disease stabilize or progress far earlier than a scan. The resolution of a scan is probably not at the level that these new blood biopsy technologies can reach. Summer, it's hard to project exactly at what time point these new technologies would replace scans, but that's certainly the promise. Some of the earlier work in blood biopsies in ALK positive lung cancer for the aficionados here had some glimmers of hope, but we're also a little disappointing about the sensitivity. Like if you have 100 patients that you know have residual disease, can you see it in every patient? And actually, the previous studies only showed that about 50% you can really see it. But there's new technologies now, and so we're going back and looking again, and we're pretty sure that these new technologies will be far better. Some are whether they'll be 70%, 80%, or 99.9%, we'll have to update you on. But certainly if it's in those upper tiers, it could replace scans in the future. That I think is exciting.
Just even hearing that that's a possibility. I mean, obviously we want those scans, right? But there, of course, anxiety comes with that. So it's exciting to hear that there could be newer and better ways, and we're limiting radiation. So that is so exciting. Yvonne, how about you? What do you?
I mean, obviously the vaccine news was super big and exciting. So thank you so much for sharing that with us this evening. And obviously we want to wait until, there's more and we can say even more about it. But coming back, I think a lot of us have been asking, and I can see Jeff in there had some other question to me. It's around, is this the Awad and Chiarli kind of protocol that we've been awaiting? Is that what's behind the vaccine that will be a part of this?
Yeah, that's a very big part of this. Roberto and Mark brought their vaccine concept to bear in this group. The earlier vaccine studies in mice are going to be a little bit different than the plan to move into humans. There's going to be more of the ALK protein used to vaccinate. The earlier studies had a few specific proteins, but it's the same concept, and it's Mark and Roberto that are really driving this forward. And in the fullness of time, there may need to be multiple types of vaccines for ALK positive patients, and we'll have to learn from those experiences to see which are the best ones. But the short answer is yes. It's built off the work that Mark and Roberto did, and that this group helped to support.
Okay, that's wonderful. There's another question here in the chat where someone's asking, are there going to be any other non-small cell lung cancer subtypes that are part of this? I'm not sure if they're referring to like other mutations, other oncogenes, or do they mean squamous versus adenocarcinoma? So
I think I put up that slide about the ultimate ambition and where do you get started. I think the first 4 years are really laser focused on using ALK positive disease as a framework to go after these smaller genotypes of lung cancer. But you could imagine, and the team does imagine, that if the framework bears itself out, you could extend it, to other subsets of lung cancer. So I think that could be in the future. Absolutely, is in the minds of the researchers. But in the beginning, we're trying to stay as focused as possible.
Okay, great.
Just to follow up while we're still on vaccines, do you envision this? Because I think this is a related question here. Is it really for patients that have active progression? Is it someone who's not progressing? Do you have any information on that?
Yeah, the inclusion criteria is still being kind of developed in real time. I think the current thinking would be for patients that have received an earlier therapy and are just beginning to show the earliest signs of progression. This new lorlatinib drug could be very effective in getting them into a residual disease state. So the idea is to use that drug for several weeks to hit residual disease, and then at that point go in with 8 or more doses of a vaccine. But we don't know exactly what all of the details will look like because it's still evolving. But that's kind of the current thinking.
Okay.
That is amazing.
Do you want me to grab the questions from the chat, or would you guys like to?
I have the next one, but I'll ask you. That's
Go
okay.
for it.
Jesse, you can feel free to jump in too. We're combining this. We saw this come up about funding. Like we have all these people collaborating together. We have a manufacturer now, fingers crossed. And there's so many different levels and components to this progress, to this project of progress. ALK Positive has committed over the next 4 years, $2 million. Wait, is that right? Yeah.
Well, over 4 years.
Over 4 years. Thanks. I'm like, wait, that didn't sound right when I said it. Over $2 million over 4 years, so 500,000 a year. Which comes from many different sources. We're so grateful for our patient community specifically that helps us to bring together those funds. But that can't be alone enough to fund a project like this. So
No, a project like this ultimately is a $15 to $20 million project over at least a 4-year period. That's pretty typical for our other team labs, and those are difficult funds for any one organization to put up on their own. And so that's where kind of a second part of the radical collaboration concept comes together. It's collaboration amongst funders and amongst funding partners to put resources together and do things together that they couldn't do individually. So Breakthrough Cancer is putting in funds, ALK Positive is putting in funds, private donors are putting in additional funds as well. We're actively continuing to develop the funding coalition with some other prominent organizations. We can update you soon as those go. And I think the work of the team and the proof of concept phase that they're in now, I think will be really valuable in solidifying funding commitments for the remainder of the funds. It's possible that biopharma could put in some funds, but as you all know, a main goal of this is to stimulate a lot of biopharma partners to get into this space, even if they believe that the space has already been addressed. And so sometimes the funding discussions can get in the way of the best science. So we think in the beginning, a lot of this is going to be philanthropy. There was a question in the chat about the current funding climate, which is really difficult, as all of you know. The level of stress and concern on the faces of basically every cancer researcher everywhere in the country is really palpable and profound. We think that this model of teamwork and team science provides a beacon of hope for the researchers actually that are feeling threatened by their longevity in kind of an uncertain moment. So that's, I think, a important social opportunity that working together can provide. Certainly, our researchers receive other grant funding, and a lot of that other grant funding can sometimes be in jeopardy, and that can change the flexibility of funds on the research side. But I think for the moment, this project is being funded through philanthropy, and that's all the more reason that we have to bring our resources together and make sure that it's not wholly dependent on what is looking to be kind of fickle sources of government support.
That's really helpful. Thank you so much. We're talking about the biorepository earlier as well, and we're just wondering, how can ALK Positive and our patient community help with this?
Yeah, I think there'll be some operational questions that we're going to have for all of you. I've personally seen that when a patient indicates that they want to donate their tissue and their data, that can sometimes break down some of the logistical barriers that will often get in the way. Each part of the project is going to have some differences. For the 9 cancer centers that are involved, they've signed a master agreement allowing for ALK samples to be shared. Breakthrough Cancer also provides program management support, and we work with project managers across the institutions, because even when the institutions have agreed, sometimes there are still bureaucratic impediments that we have to help to support the scientists to work through. But luckily now this is not our first rodeo. This is our ninth rodeo. And so we're learning as we go. And we figured out what some of these impediments are. So it's not seamless, but we're trying to get it as seamless as possible for the 9 cancer centers. For other tissue donation outside of those centers, some will come through the direct-to-patient framework that I mentioned, and we'll be in touch with you guys as that launches. And then in other instances, there may need to be some hand-to-hand combat with individual institutions or individual biobanks to free up initial samples. It's our experience that kind of everyone actually wants to share. It's just, I think, very few scientists and very few organizations are well equipped to navigate through the complicated legal and operational dynamics that often get in the way. And as I said before, we're getting better and better at that as we go.
No, and that, do you see that question that came in too? Like if their cancer institute won't sign the MTA, would they? And you're probably still working on all of that logistics.
Yeah, happy to, it's sometimes hard to answer these questions individually. I think uh, I would not assume that you have to travel to one of the 9 cancer centers to participate. I would get in touch, and you can look at our website or the ALK Positive website, I'm sure, and identify the research teams that are involved. I would get in touch with one of them and say, hey, I'm from X institution. I'd love for my samples to be included. What do you think? Christine Lovely or whoever, Roberto Chiarle, what do you think is the best way for my tissue to get involved? And I think if the researchers can provide guidance, then we can help to facilitate it. In some instances, yes, it might be important to travel to a cancer center, but that wouldn't be my first guess of how to participate.
Wonderful. In the chat, we see a couple people interested in understanding, will the vaccine only be available to those already on lorlatinib?
No, it would be available initially, and as I mentioned, the trial details are still being refined in real time. The plan would be patients that have received a different ALK inhibitor, but not lorlatinib, because we want to make sure that the lorlatinib drug can drive patients into the residual disease state. That isn't to say that other trial cohorts couldn't be added in the future. And as I showed on that slide, we hope the vaccine trial is just one of many trials. So we'll continue to update this community. It's our hope that in the fullness of time that, basically all patients that can get into residual disease could be eligible for one or more trials, but you have to start somewhere.
Great. And Jesse, this might be a difficult question to answer, but I live in London. We serve a worldwide group. We have members of our community around the world that are super excited about this project. Do you ever envision a time where other institutions around the world might join this as well? Or how can other patients from other places outside the US join potentially?
Absolutely. I think scientifically, there's no prohibition on collaborating and working together. I think, as you're well aware, there are a couple of procedural hurdles for international tissue donation related to some of the European guidelines. Uh, and but there's no reason we couldn't work through those if there was the scientific opportunity was significant enough. But with that being said, you might imagine that we're first trying to get these 9 institutions for that to work seamlessly. So we've learned a little bit. If you try to expand too quickly, too fast, you can undermine this kind of spirit of teamwork that really needs to be established. And once it's established, it becomes a bit easier to add others to it. It's actually, as many of you might recognize who are involved with teams at work or at home or somewhere else, it's hard to work as part of a team. You have to listen, you have to collaborate, you have to compromise. And some of the scientists are better at that than others. So sometimes scientists say, oh, I'd love to be involved. How do I get involved? Thinking of the funding opportunity, not really knowing how hard the sweat equity is to disagree about something and work to compromise. So it just takes some time. We actually often find our younger researchers are more passionate and able to work together in this new way. Some of the more established researchers do this well, but some of them have, operated a certain way for a very long time. And, like many of us that are a little older, it's sometimes a little hard to change our ways. I wasn't mentioning anybody in particular. Just,
We hear you. We hear you. And I think, yeah, let's get the project started. But it's great to hear that that's an ambition and an interest as well.
Yes, and we have lots of vaccine questions still coming in. I think the biggest thing is that the protocol is evolving and not yet set. And it could end up with patients that have only been on lorlatinib or whichever, I know, but it's evolving.
yes.
Yeah.
I received the latest draft of the protocol last night. So these things are evolving in real time. So it'd be premature to be definitive about these things, but we'll work collectively with ALK Positive to make sure that you guys have the most up-to-date information possible.
Fabulous. And that repository, is that blood and tissue, or is it just tissue? What are you going to be collecting?
The direct-to-patient mechanism is best set up for blood and for data, or for stored tissue that might be in a freezer somewhere. To collect residual disease tissue via direct-to-patient is pretty hard because that tissue isn't always sampled. So the plan is to get blood and frozen tissue and data via direct-to-patient, and initially to focus on going back and getting this residual disease tissue at the 9 cancer centers. But, Summer, you asked the question about blood replacing scans. Another exciting thing would be blood could replace the tissue biopsy itself. And if we can do that, then you could, go and collect blood pretty seamlessly.
And even so, for someone like myself, who initially 5 years ago, and this is showing again how times have changed, the liquid biopsy was around then. And I'm just using this as an example, sorry. But I had both, but the ALK was not picked up. So this again in the blood, yes. So I had to wait for my tissue sample. So it's exciting to hear that this is even so much more sensitive that you could rely on that.
Could be. I think for the diagnostic sample, initially when you're diagnosed, it's hard to imagine not having the tissue at that moment.
Yeah.
What I was referring to is as the disease continues,
Okay.
especially in this residual stage, if there's biology that we need to measure in the residual disease stage, we might not have to subject the patient to another procedure. We might learn that we can just take bloods all the way through. It's possible that blood could replace all tissue sampling, but
That's the first step is
probable that we're going to, especially for diagnostic biopsies, still want those.
Okay, great. There is another question that just came in. Hold on.
Well, the University of Michigan and their study
yeah.
partner.
We've had productive conversations. There's a lot of mutual interest with Michigan and with other institutions. We're obviously just getting going. So I think what everyone is saying is, let's get everything going. Let's find the opportunities and work together. But I wouldn't want anyone to come away feeling like this is only about these 9 institutions or only about 5 institutions. This is just trying to build a collaborative community to make data, produce resources that everyone can benefit from. But we try to be very science focused, like what is the science that's needed right now? And if that science requires new interactions, let's go for it. If the science requires making data and posting it on the web really fast so that everyone can get it, then that's the best approach. So we try to be very practical about the science. But yeah, the Michigan team and others were in active conversations with.
Great. Okay, and then finally, I think we touched on this a little bit in earlier, but that minimal residual disease state, can you just give us a quick little recap again?
It's actually a deceptively complicated question, even asked very simply. And actually, this is something the team is working on, and in some cases struggling, because each institution defines this a little bit differently, and each clinician defines it a little bit differently. So even though it sounds very simple, I think one of the outcomes is going to be a much better answer to Deborah's question, with the consensus definition of residual disease. Right now, they're defining it as disease that's left over after an initial strong response. It can be macroscopically visualizable, meaning like you can see it on a scan, like I showed you before, or it could be microscopically visible only, where the scan looks clean, but the blood biopsy test can identify it. And so one of the things the team is trying to do is to integrate information from the scans, from blood biopsy, and from other criteria to build a consensus kind of staging model of residual disease. So I don't know if Deborah intended for it to be a tricky question, but it is one.
I was just going to thank her. Deborah, thanks for asking the tough questions.
But yeah, I think we'll learn. It may be the size is most important. It may be the biology is most important. It may be the velocity of initial response is the most important. These are all things that we need to work on. But rather than working on them like one institution at a time, the idea is to work on them together.
Great. Hold on here.
Yeah.
Wait, that just popped up. How about the frozen tissue or tissue from a while ago? Is that what you're thinking that you would want? Like our wedges, our tissue and wax wedges are from our initial diagnosis?
Yeah, it's called formalin fixed paraffin embedded, the FFPE tissue. Yes, I think that type of tissue could be very valuable. It'd be good to get in touch with one of the researchers involved, and we or ALK Positive can facilitate that. Um, there are other great initiatives. People mentioned the University of Michigan initiative. There are lots of great initiatives to donate tissue to, and so I also wouldn't want anyone to feel like they have to donate it to this initiative. I would definitely try to make sure that your tissue and data is being utilized by ideally multiple open source initiatives that are trying to make data and put it into the public domain. And so happy to talk to anybody or to any institution about whether this is the best place for it versus something else.
Okay, awesome. Thank you. Do you have another question or you want to?
I think we can, there was one more question just asking, can ALK positive disease be diagnosed in the blood yet? And I think someone did reply. I don't know if you want to add anything to that, Jesse.
I don't think I'm the right person to answer that question. That's more of a clinician question.
Wonderful.
And we have seen in our community, and for many of you can attest that through a blood biopsy, you might have received your diagnosis that way. And that was my explanation
Yeah.
earlier, saying that for me, that didn't work because my tumor burden wasn't enough. I wasn't shedding.
Stopped actually.
And
so
see?
I got mine faster that way. My tissue was taking forever and the blood came back like in 10 days.
So some yes and some no. So good call, Jesse.
Yeah.
Pass that off.
Exactly. But I think we can turn it over.
Yeah, this has been so great. Thank you. Kirk, would you like to say a few words, please?
Yeah. Can you hear me, Summer? Yeah. First, I want to say thanks to Jesse for being here on a Sunday night. I've had the chance to hear Jesse speak a number of times, and I'm always come away motivated and inspired in what's happening with Breakthrough, and he does such a great job and is so willing to be a part of this. So we really appreciate that, Jesse. I had 3 points I wanted to make real quick just from some of the presentation. One of them that I think for this group that's watching to know is Jesse listed a lot of the researchers that are involved with this this project. I hope you'll note that a good number of them serve on our scientific advisory board, and we as Summer and Yvonne mentioned, we had a working dinner with them with that scientific advisory board on Friday evening at ASCO, and those are, and I don't have the full list, but some of those that are part of that are Dr. Awad, Vincent Lamb, Camage, John Haymack, Jessica Lynn. And Jessica Lynn will be our keynote speaker at the summit this July in San Diego. So she will probably talk a little bit in that keynote. I don't know for sure, but she will probably cover a little bit about Breakthrough Cancer Project. I also wanted to mention that there was a little talk on the funding earlier, and I hope all of you remember when we had this last year with Jesse, it was a kickoff of ALK Positive Awareness Month, and we were kicking off a fund drive to get our initial funding, which was a $500,000 funding for the kickoff the Breakthrough Cancer Project. And we hit that mark, I think, less than 3 weeks into the month. And that was all because of y'all. People like you that were doing this. This is a very much a patient-driven collaborative project. And even to make an even bigger point of that, I think is the last point I wanted to make, is the reason we even have this collaboration, and Jesse, correct me if I'm overspeaking or quite incorrect, but most of this comes from BTC already existed, and you saw in Jesse's earlier presentation, it came about by those other 4 cancers that were difficult to treat diseases. but the work of some of our ALK patients is what brought us to BTC's attention. And I think that's important to mention. I'm looking at one of them down here on my screen. And I see Emily Venanzi, who is one of those big heroes. Emily, Mark Muscovich. There are a few others that I probably don't want to be named yet, so I won't list their names. But Emily and Mark Muscovich are huge parts of this. They're leaders on our medical committee. They're two of the smartest, most humble people you could ever meet. And Emily is a big part of why this happened. And I think to me that's so important to share, to emphasize how important the patient voice is. A lot of times we know that sharing our stories is important, but there is groundwork here like this that's happened because of patients that got out there and were not sitting on the sidelines, but decided to make things happen. And Emily's been a driver in our medical committee since she was almost since the month she was diagnosed. And I think that was around 2018, 2019, if I'm correct, Emily. So, Emily, I don't know if you have anything you want to say. I don't want to put you on the spot by surprise, but you may have something you want to add to this.
Well, thanks a lot, Kirk and Jesse, for being here tonight, especially during ASCO. It was definitely a team effort among us ALK patients to make the connection with BTC, and we were really impressed with what they were doing, bringing institutions together for the work on other cancers. And as ALK Positive, we had been speaking individually with all of these great researchers at these wonderful institutions. But at the same time, we're saying, what if they could work together even more? And then we realized, BTC is doing exactly this. So we thought it was really a perfect fit for ALK Positive to get involved. And we were so pleased when they agreed to help us and form the lung team and work together with us and all the lung researchers. So just wanted to express my appreciation as well to Jesse and to the BTC team for.
And fantastic, Emily, to work with you and Kirk and others. And Kirk, you're exactly right. I think this was not a cancer type on our radar when we first launched, and you guys and Emily and Mark and others really elevated our attention in an area that, of course, you were so passionate about.
Kirk, you're on mute.
I was going to say, just keep that in mind that you have power as patients. So remember that. And I know that Katie Delaney has something she wants to add about ALK Positive Awareness Month.
I'm honored to be with all of you tonight, especially on the first day of ALK Positive Awareness Month. I think throughout this entire presentation and from the voices of the patients themselves, you can see that one message rings true throughout, and that this is what patient-powered change looks like. So throughout the month of June, we are asking all of you to join us in raising awareness and funds for our Powered by Patients campaign. As you can see on the left-hand side, we have these Powered by Patient cards highlighting you or a care partner and what powers you through this diagnosis. Don't worry, it's not too late to participate. If you would like to join us, head to our website at www.alkpositive.org and select the Ways to Give button. Here you'll see how you can participate by either creating a card, creating your fundraising page, or learning more about this campaign. That's not it. We do have another $500,000 goal this year, and with the incredible generosity of some matching donors, every gift donated throughout the month of June will be doubled up to $262,000. This doesn't only double your dollar, but it doubles your impact and gives us twice the chance at a future without ALK positive cancer. So throughout June, I hope you'll remember that every research project you see on our ALK Talks, the new treatment options emerging, and the momentum towards a cure wouldn't be possible without you. Your voice fuels research, your story shapes science, and your strength drives change. So let's show the world what being powered by patients truly means.
Fabulous. Thank you so much, Katie. It is so true. ALK Positive is a patient-driven organization. Our community is what fuels the organization. It's powered by patients and for patients. So thank you all so much. Thank you, Jesse, for being here on a Sunday evening with us. And thank you to Emily and those who have made this possible. Thank you, Kirk and Nancy, for joining in as soon as you landed from ASCO. Thank you to Mark, Dana, and Katie for keeping this going on your end, and to my sidekick, Yvonne Diaz.
It's been
Thank
great joining
you.
you here together.
It's been so great. We can't wait to see so many of you in Summit. Oh, and Sadia, thank you, Sadia, too, for joining us from your hotel room at Summit. We all can't wait to see so many of you at Summit in just almost a month, right? It's June today, so
Yeah.
next month.
Wow.
We do have another ALK talk later this month with Stephen Liu that will be talking about progression. And all sorts of other things. So be sure to join us then. We'll also have coming up like a Know Before You Go for Summit, whether online or, whether you're attending virtually or in person. So we'll be excited to bring that to you also. Remember, we have a stacked calendar of Healing Arts Connect, Find Your People. Even if you come once every few months, it just might give you that little support you need. So it's there for you when you need it. And if you would all like to unmute and say thank you as well, that would be great. Thank you all so much for joining us.
Thank
We'll
you.
see
Thank
Thank
you.
you.
Thank
you.
Have a great night.
Thank you, everyone.
Bye. Bye.
Thank you.
Thank you.
Yes, thank you.